Dear friends, yes, the fate of your brain is truly in your hands, and we're going to explain why. We present some facts here. Objective facts, scientific facts. Some are part of global medical research accumulated over more than 50 years. Others result from our own observations, which we have published in highly regarded scientific journals.
Alas, we are obliged to tell you that the findings are quite alarming, hence this video. But since our following message may be useful, since our message can help you better manage, optimize, and protect your brain, it seemed necessary, even vital, to inform you. This particularly concerns the youngest among us, but for others, it may not be too late.
What can we say about the state of our brain? It all depends on age. How does our brain age? Scientific progress, combined with increasingly sophisticated observation techniques, not to mention the accumulation of international scientific data, allows us to describe brain aging over the next 100 to 120 years. It is thus possible to determine its strengths and weaknesses, its robustness and its vulnerabilities. And we will describe its weak points. Our brain is doubly protected. First, behind the skull, and second, by the blood-brain barrier. This barrier filters compounds in the blood before they can access nervous tissue. Because of this, is the brain relatively spared from the diseases that affect other organs? One can easily imagine that brain pathologies would affect intellectual performance. Some may be subtle, but others could be catastrophic and lead to dementia. What is the reality of the situation? What do epidemiological studies show?
International clinical statistics indicate rather catastrophic figures. In fact, this assessment shows that dementia, meaning the total loss of intellectual functions, affects 1/3 of patients aged 90 and 1 out of 10 patients at age 80. This dementia can be of the Alzheimer's type, a degenerative process, vascular, or a combination of both. What about Alzheimer's disease specifically? Regarding the prevalence of Alzheimer's disease, the figures are equally impressive and alarming. Just imagine, according to studies, approximately 30% of patients aged 90 are affected. Similarly, about 10% of the population aged 80 is affected by Alzheimer's disease. Globally, and with the aging population, the number of Alzheimer's patients is considerable. This figure is estimated to be around 50 million patients. The same is true for the astronomical public health costs, because there is still no effective treatment. The diagram presented here shows that the critical age for the onset of Alzheimer's disease is 65. Almost 2% of the population age 65 is affected. Then, the prevalence of this disease increases exponentially. What happens at this critical age of 65? That seems to open the door to the onset of the Alzheimer-type degenerative process. To answer this question, we must analyze the dysfunctions of the Alzheimer's degenerative process at the molecular level. We know that 2 pathological mechanisms synergize to cause Alzheimer's disease. One, linked to the APP protein, for Amyloid Precursor Protein, and the other, linked to the aggregation of tau proteins. Indeed, the abnormal fibrils that accumulate in degenerating neurons during Alzheimer's disease result from the aggregation of pathological tau proteins. By quantifying pathological tau proteins, shown here in grayscale, we can track the progression of the brain's degenerative process in Alzheimer's disease. 10 stages of invasion of the degenerative process are distinguished. The disease progresses gradually from relatively asymptomatic hippocampal stages 1 to 3 to severe dementia in the final stage. It is important to note that the degenerative process begins specifically in the hippocampal region, the seat of memory. This postmortem study was conducted on a cohort of over 200 cases, ranging from non-demented controls to patients at various stages of Alzheimer's disease. For each case, each Brodmann area of the brain was analyzed for its content of pathological tau proteins. The technique used was immunoblotting. We can thus see the progression of the degenerative process from the hippocampus to the temporal cortex, then the associative areas of the frontal, temporal, and parietal cortex, then the motor and sensory areas to invade the occipital cortex, generally at the very last stage. Thus, our postmortem study on non-demented patients, that is, from the general population, highlights the vulnerability of the hippocampal region to the neurofibrillary degeneration process. In other words, pathological tau protein aggregates are systematically observed, though at low levels, in all individuals aged 65. This phenomenon becomes much more significant and consistent in individuals aged 75. In this age range between 65 and 75, the presence of pathological tau proteins is not correlated with significant cognitive impairment. However, there is no evidence that it is not correlated with milder cognitive impairments, which may be revealed by more sensitive cognitive tests. This is what we will see in the following slides. To better study this hippocampal vulnerability, brain imaging can provide valuable information. MRI, magnetic resonance imaging, is now a routine tool in university hospitals. Moreover, the examination is quick and minimally invasive. We were thus able to conduct a study called SUCCEED on asymptomatic and apparently healthy volunteer subjects aged 50 and over, average age 64, followed at an outpatient clinic. This study allowed us to quantify white matter lesions using the Physicus scale, as well as the degree of hippocampal atrophy using the Shelton's scale. Magnetic resonance imaging allows visualization of white matter abnormalities. White matter consists of bundles of nerve tracts in which each axonal fiber is surrounded by a myelin sheath. MRI visualizes these vascular alterations of the white matter, known as vascular leukoencephalopathy. The disappearance of myelin sheaths can then be observed. Several levels of severity are distinguished, quantified by a scale proposed by Physicus in 1987. There are 3 levels of severity of vascular leukoencephalopathy, shown here. Analysis of our cohort, reflecting the general population aged between 55 and 65, shows concerning results. Nearly a quarter of participants, 26%, present an MRI with a Physicus scale score of Stage 2. This Stage 2 is far from insignificant, as it indicates significant damage to nervous tissue, resulting from neuronal loss or loss of connectivity between nerve cells. We conclude that the pathological phenomenon of vascular leukoencephalopathy is common in individuals over the age of 55. We have seen that the hippocampus is vulnerable to tau pathology during brain aging. It was possible to study hippocampal atrophy, a sign of its vulnerability, in our cohort of individuals aged 55 to 65. This is possible through a simple MRI examination. This atrophy can be quantified using the Shelton's scale. There are 5 stages, the first with no atrophy, and the last with severe atrophy, known as Stage 4. What are the results for our cohort? We found that 17% of individuals in our cohort present a significant degree of hippocampal atrophy, Shelton's greater than or equal to Stage 2. This confirms the fragility and vulnerability of the hippocampus in individuals over 55. Furthermore, we observed that the degree of hippocampal atrophy is correlated with the amount of white matter lesions in these participants. In conclusion regarding our imaging observations, we note 2 essential points. Firstly, White matter lesions and hippocampal atrophy are common and frequent in the general population of asymptomatic individuals aged 50 and over. Secondly, the frequent vascular damage in individuals over 50, visualized by white matter lesions, could explain the vulnerability of the hippocampus, a stepping stone to Alzheimer's disease. This can be explained by the hypermetabolism of the hippocampal nerve cells, which are involved in all brain activities, particularly in managing short-term memory. In conclusion regarding our imaging observations, we note two essential points. Following these observations, we logically asked ourselves the following question. Do these brain signatures, both molecular and anatomical, have an impact on cognitive functions? To answer this, we performed cognitive tests on these same patients. First of all, what are these tests? First, we used the clock drawing test, which is well known and highly sensitive. The patient is asked to draw a clock and indicate a time, here 11:10 in our example. Depending on the quality of the result, a score is given. This test allows us to assess executive and visuospatial processes. The results from our cohort indicate that nearly a quarter of participants show impaired cognitive functions on the clock drawing test. This result is both surprising and concerning. As part of our SUCCEED study, we were able to assess our cohort with more comprehensive cognitive tests, such as the FAB test described here. FAB stands for Frontal Assessment Battery. This test assesses conceptualization, verbal fluency, sensitivity to interference, motor planning, inhibitory control, and environmental autonomy. The results obtained using the FAB cognitive test are as follows. More than 1/3 of the studied subpopulation, aged 50 to 65, show executive dysfunctions on the clock drawing test and the FAB. Other complementary cognitive tests, such as the MMS, Mini Mental Test, which includes 30 items to examine global cognitive functions, or the Dubois 5-word test, which assesses episodic memory, point in the same direction, namely, participants showing a deficit on one or more of these tests have, statistically speaking, white matter damage and more or less severe hippocampal atrophy. Inversely, individuals with white matter and hippocampal abnormalities visible on MRI have a more or less significant cognitive deficit. In conclusion, these approaches make it easy to identify the at-risk population, opening the way to an effective preventive therapeutic approach. The overall finding from studying the human brain in all its forms, whether clinical, molecular, through imaging, or through cognitive tests, leads to the same conclusion. The first is that the brain is a fragile organ that shows clear vulnerabilities as early as age 55. The second is that these significant, frequent vulnerabilities point to a path of prevention. This path logically involves advocating a healthy lifestyle, even though our hectic lives never seem to leave us time to follow such good advice. And of course, to implement this very reasonable program as early as possible. What could this program look like? How to protect and properly maintain our brain? Taking care of your brain is a bit like maintaining a Formula One car. It requires the right settings, the right fuel, and above all, not staying in the garage too long. Here are the essential pillars to boost your brain plasticity and protect your cognitive capital in the long term. Let me introduce a very simple idea, but one that has a powerful impact on your brain over time. The 5S rule. 5 pillars, 5 daily habits, 5 levers you actually control. First, sleep. This is when your brain does its maintenance work. It cleans itself and consolidates everything you've learned during the day. Second, physical activity. Movement is not optional. It's fuel for your brain. Better blood flow, less stress, stronger neurons. Third, cognitive stimulation. Your brain needs challenge, not routine, challenge. Learning something new is what keeps it alive and adaptable. Fourth, social engagement. We are not meant to function alone. Interaction stimulates multiple brain systems at the same time. Isolation does the opposite. And finally, sustenance. What you eat is quite literally what builds your brain. Quality fuel matters. And here is the key message. These 5 elements don't work separately. They amplify each other. Together, they create real cognitive resilience, and they directly influence how your brain will age. Sleep is not a passive state. It's when the brain does some of its most important work. During the night, a dedicated system called the glymphatic system acts like a cleaning crew, removing toxic waste that builds up during the day. At the same time, the brain is organizing and storing information, strengthening what you've learned and experienced. In other words, sleep is not just rest, it's maintenance and optimization for your brain. Movement is not just good for your body, it's essential for your brain. When you move, you increase blood flow and oxygen supply to the brain. You also reduce stress, one of the main enemies of memory. And importantly, physical activity stimulates the production of BDNF, a protein that acts like fertilizer for your neurons. In simple terms, exercise helps your brain grow, adapt, and resist decline. Your brain follows one simple rule: use it or lose it. But repetition alone is not enough. What truly strengthens the brain is novelty and challenge. Learning a new skill, a language, or even changing your routine forces the brain to adapt. That's how you build neuroplasticity, your brain's ability to rewire and stay flexible. We are not designed to function alone. A simple conversation activates memory, language, attention, and emotional processing, all at once. On the other hand, isolation significantly increases the risk of cognitive decline, and stress plays a major role. Chronic stress releases cortisol, which can damage key brain areas over time. That's why connection and stress management are essential for brain health. Your brain is built from what you eat. Food is not just energy. It's also the raw material your body uses to rebuild itself every day. High-quality diets, such as Mediterranean or Okinawan diets, are strongly associated with better cognitive health. Think of it this way: you cannot build a strong structure with poor materials. Good nutrition is foundational to long-term brain resilience. At first glance, everything we've said may sound obvious. Sleep well, move more, stay engaged, eat properly. Nothing new, right? But here's the reality. Obvious does not mean optional. Because if these principles are ignored over time, there is a price to pay, and it is often paid later through cognitive decline. What science shows us is very clear. Your brain has natural vulnerabilities. They are part of aging. You cannot completely avoid them. But what you can do is influence how and when they appear. That's where the 5S rule comes in. It's not about perfection. It's about consistency. Because these 5 pillars, sleep, movement, stimulation, social connection, and sustenance, don't just add up. They amplify each other. And together, they can significantly protect your brain over time and even improve your overall well-being and mental balance. Thank you for your attention. If there's one thing to remember from this presentation, it's this: your brain is not just something you're given, it's something you actively shape over time. And the good news is, you already have the tools: sleep, movement, stimulation, connection, nutrition. Small actions repeated consistently can make a significant difference. So now I'd be happy to take your questions.